We're live. Everybody say hello.
Hi.
See? There you go. Every video begins with me dancing my hand back. So, that was the lesson we covered, was protein structure issues, and so we've been covering, protein structure, kind of hammering away at that because of its criticality in terms of how proteins work and how the rest of the discussion we're going to be carrying out will go on in terms of using proteins to do things. And so we finished up with a couple of diseases that are involved in protein folding, and things like mad cow disease and Alzheimer's and Parkinson's. Mad cow particularly is weird because the shape is actually a malformed shape that is disease-causing, and if it bumps into a normally shaped protein, it can autocatalytically shift it into the disease-forming shape, basically making it insoluble. So mad cow is transmissible. Alzheimer's and Parkinson's doesn't seem to be. The transmission in humans of this was, it slowly came about, but one of the key points was a tribe in Papua New Guinea that tended to eat their dead, which I don't judge. The women would be given the soft parts like the brain, whereas the men would take the heart and muscle. Makes sense. And women and children would preferentially begin developing this disease they know as Kuru. And so that was what led to the idea of the transmissibility from eating brain. So let that be a lesson to us. Really, don't eat brains. They used to actually, my dad used to talk about eating calves' brains, and I'm like, why? I just will have a bowl of cereal, thank you. But we don't eat brains or nervous system much anymore. It's typically off the table, as it were. But if you're going to be a cannibal, go ahead and choose the heart, skeletal muscle, make up some story that, that's how you go with it. Don't eat brain. So there is now in deer something called chronic wasting disease. And guys that are deer hunters know of this. You actually have to take your deer to the DNR check station, and they do a cursory analysis of it. And mostly, if you're not eating nervous system tissue, you're probably pretty good. But there is chances of getting contamination. Cross species is pretty hard to do, but it does happen. And so chronic wasting is a bit of an issue for deer hunters. Again, I think I'll just have cereal. Thanks. So we're going to skip over chapter 5 and 6, which is dealing with DNA replication and DNA repair, mainly because we covered this in genetics. I have other things to talk about. I never covered DNA replication. We get into a little bit more of DNA repair and mismatch repair and recombination in molecular genetics. So if you're interested in that, there's some very cool things going on these days with a system known as CRISPR-Cas9, which maybe you've heard of, which is a bacteria virus system that allows for a swap out of nucleotides. You provide an RNA template, and the Cas9 enzyme can target that spot in the genome and actually rearrange and change the nucleotide sequence in that exact spot. And that presents some pretty cool tricks for being able to manipulate organisms, because it turns out this system works in eukaryotes. So we actually did some experiments upstairs using bacteria, and it worked. Really what we did with bacteria is we made them resistant to an antibiotic, which it didn't really make anything jump out. It was just that the bacteria grew. But the possibility of doing this on embryos is a pretty cool thing, in that you could actually change specific nucleotides and you could alter protein function that way. Okay, so that was DNA recombination. And DNA replication, yep, DNA replicates, and we have chromosomes. But what we want to get into is what is known as the fundamental or basic dogma of molecular genetics, and that is the flow of information from DNA to RNA to protein. And this is a really fundamental point, and so you need to kind of get all the terms locked down pretty good and the overall flow of things. This, of course, step between DNA and RNA is known as transcription, and this step is translation. And so we talk a lot about post-transcriptional regulation and modification, post-translational regulation, post-translational modifications. And so knowing your way around the steps here is a critical point, and being dead set on the terms. DNA is transcribed into RNA, RNA is translated into protein. So, there's a lot of philosophical discussions about this mechanism. Part of it is that DNA is a very stable molecule. And every cell in your body has its DNA template. And it's pretty much identical. I mean, we were discussing in MCAT review the immune system, and the cells that produce antibodies actually do go through a chromosomal rearrangement to produce the functional antibody genes. But for pretty much all of your cells, the genomic DNA in one cell is more or less the same as the genomic DNA in any other cell. I mean, there are all kinds of weird exceptions, but that's a fairly good, true statement. So what we have then is the coding potential in every cell for every possible protein. Then the regulation comes in as to how a specific part of the DNA, what we call a gene, is going to be transcribed. And the advantage of this is we can have, this is a sort of a cartoony diagram here, gene A and gene B, and we can regulate the amount of product by regulating the amount of RNA. So we might have a whole bunch of RNA being produced from gene A, and only 1 or 2 RNAs being produced from gene B. So this is a standard kind of phenomenon, in that depending on the cell type, there are sets of genes that we call the housekeeping genes. There are little people with brooms and brushes, and they come around and polish up your cells. These are the genes that are typically expressed in every cell type at the same level. And from an actual molecular genetics perspective, finding a single gene that is expressed in all cell types at the same level is theoretically okay. An enzyme involved in glycolysis, or an enzyme involved in some kind of glucose metabolism should do it. There's actually some variance going on there, and so you have to take that into account when you're doing experiments where you're trying to quantify the amount of RNA from a single gene. But again, from a most logical perspective, we've got genes that are expressed in every single cell, and then some genes are expressed only in a single cell, or only during a very narrow window of development. And so there is a wide range of gene expression going on. And DNA provides the ability to have every cell have the template, but not necessarily require to express every single gene. And so we can have cells that express a subset of genes, and another cell that expresses a different subset of genes. And this is the way it works. You can envision other control mechanisms, but that ain't how we do it. When we find life on Mars, well, we'll probably find they do the same thing. But this is one of the notions about how we can think about the utility of having a DNA template and then being able to produce RNA from it. So a couple of the players involved, just to refresh, we've got the nucleotides, and the key difference between RNA and DNA, we have the base, and then we have the carbons on the ring structure of the 5-carbon sugar, give the prime, so it's 1 prime, 2 prime, 3 prime, 4 prime, and then the 5 prime sticking up, and then we have the phosphates sticking out here. The 2 prime may or may not have a hydroxyl. The 3 prime absolutely has to have a hydroxyl. And so we are always referring to DNA in the 5' to 3' direction, or talking about the 5' phosphate and the 3' hydroxyl. And that's because when we're going to make a polymerizing a nucleotide chain, we have to have the 3' hydroxyl as the acceptor. So we've got the 5' phosphate here of the next nucleotide. There's the base. And we have OH. We have OH. We're going to make the phosphodiester bond where we take and link up the phosphate to that oxygen. That's a critical point. If you don't have a free 3' hydroxyl, you can't add the next nucleotide. And this orientation is important and useful when we start to think about some of the structures, because if you always remember, you got to have a free 3' hydroxyl to elongate the chain. That allows you to start to think about the order and the sort of the organization of how we're going to be building a new polymer. Dramatic pause for coffee. So as we've been saying before, when we have a 2' hydroxyl, this is the fully oxygenated sugar. This is the ribose sugar. So the ribose sugar, look for the 2' hydroxyl. If we remove the 2', we deoxygenate it, and that is the deoxy ribose. So the 2' carbon has simply a hydrogen on it. And as I'm writing this occasionally happens to me, I'll have this thought of, I wonder. From a chemical perspective, I do know that RNA is not very sensitive to acid. And you actually can preferentially purify RNA by making the extraction solution significantly more acidic. DNA is broken up in the presence of strong acid. DNA is perfectly fine in the presence of 2 or 3 molar sodium hydroxide, so base doesn't bother it, but acid does. And so that's not entirely what makes DNA is much more stable, and RNA with its second little hydroxyl, RNA is unstable primarily because of the presence of a whole range of enzymes that are collectively known as RNases. RNase is what's going to be degrading RNA. And I think this is primarily a fallback from the system, in that if you've got RNA floating around, you really are only one step away from turning that into a protein. And depending on what kind of cell you are and what the RNA is, that protein might do something that you don't want it, you don't need to have happen, like it will start triggering cell division. It could cause some kind of cell signaling event. So we have to keep a really tight rein on RNAs. RNAs that shouldn't be around, they get degraded very, very quickly. And because of this, then RNases are very tough proteins. They're floating around even in the dust. And so simply touching a tube that has RNA in it can introduce RNases that will completely degrade your RNA. So purifying RNA is a bit of a trick. Purifying DNA is a piece of cake. All you got to do is grind it up and boil it, spin down the pellet, and you probably will have a reasonable amount of DNA. One of the experiments that we used to do sort of, pre-COVID on campus visit day, and some of you may have been victims of this, we isolate DNA from a strawberry just to play around with it. And I've always been a little curious about this, in that it's really easy to do. You take a little bit of detergent, like dish soap, mix it with a pinch of salt, and make an extraction buffer. You squish the strawberry up in a Ziploc bag, filter it through paper towel, and you end up with strawberry juice. You add straight ethanol to it, and you get this tremendous blob of white strands starting to come out of solution. And that's a lot of DNA, and I've always suspected that actually some of that is starch. But you can twirl it out and get this big old snotty glob of DNA and put it into an Eppendorf tube and take it home. whether it's actually totally pure DNA. I've actually done a little bit of messing around with it, and it doesn't seem like it is, but it looks really cool. So we can extract DNA, though, from a lot of different tissues pretty easily. RNA just is a lot harder to do. I mean, you can definitely extract RNA, but RNA is just a harder thing to mess with. RNA being single-stranded, you can't clone it, so you can't actually incorporate it into bacteria and have the bacteria replicate it, which in the molecular biology biz, when we talk about cloning things, rather than making little people, really what we're doing is talking about taking bacteria and using them to produce a given chunk of DNA, so we can clone that DNA. We can generate DNA using the polymerase chain reaction, which we'll talk a little bit more about and do some stuff in lab. Just as a thought, we still aren't meeting in the lab this week. I think we're going to probably start meeting up next week, but I'll let you know. So DNA is a fairly tough molecule. RNA we tend to think of as being a fairly delicate guy. And the only difference is the hydroxyl here. You've got to have that hydroxyl. All right, so we've got then this orientation of the DNA. And this is another real critical kind of diagram concept that is a little hard to initially get, but you just have to start working with it and kind of think about it for a while. When we draw out a DNA molecule, generally you just draw a line. All right, we don't need to get real creative. And when we think about the DNA itself, it is in fact double-stranded. And so we've got the 5' end and the 3' end. The opposite strand, the complementary strand, is running in the opposite direction, 3' to 5'. So that's the way we diagram it and think about it. Now, when we're talking about DNA sequence, and we'll be doing some DNA sequence analysis, when you're given a sequence, it's like GAATTCATGCAA, just a sequence. It is assumed that when you're given a sequence, it is written in the 5' to 3' direction. That's just the way you do it. Otherwise, you're being a nerd. All right. Now, that means that if you're given the top strand, because we know how to complement the nucleotides, you're a very good nucleotide, and we know how to base pair them, we can actually come up with the bottom strand. So the bottom strand is generally never, ever given to you. It's confusing. We don't do it. So the bottom strand for this would be C, T, T, A, A, G, T, A, C, G, T, T. Yeah? All right. Now, here's where it gets a little bit tricky upside down. This is the 5' to 3' direction. This is running the 3' to 5' direction. When you're given a piece of DNA sequence, unless you are explicitly told, here's the gene and this is the direction that it's running from, and generally when you're given that, you'll have a little arrow that'll say, okay, the gene runs that direction. Because genes don't all run in the same direction. You can have this gene being transcribed that way. And so when you're given a sequence, it's the top strand, but there is no reason to think that the gene that is transcribed from that piece of DNA is running in that direction. And so when you want to actually look at both strands of DNA, and there's going to be a homework assignment that I'm going to post up to Moodle that will be doing this, for the bottom strand, you actually have to write it backwards. So just as a general note, when you're given the top strand, you can base pair and find the bottom strand, but in order to actually look at the bottom strand, in a sense, as if it were the top strand, or to do any manipulations with the bottom strand, you got to write it in the 5' to 3' direction, i.e., you have to write it backwards. So this would become T, T, G, C, A, T, G, A, T, T. 5' to 3'. So that is the deconvolution you have to do with the genetic code when you're monkeying around with it. The top strand, you just read it. You're good. The bottom strand, if you want to actually think of it going the other direction, you got a base pair and then write it backwards. We'll get into this a little bit later, but I thought I'd throw it out here now just to confuse you. Yay. All right. So this is the top strand. Now, when we think about taking DNA and turning it into an RNA molecule, what is the enzyme we use? I mean, it is somewhat rhetorical, but hopefully you recall this from genetics. It is the enzyme RNA polymerase. RNA polymerase always works to build the new nucleotide chain in the 5' to 3' direction, i.e., RNA polymerase is going to make, in RNA form, the top strand. So if we were to take this chunk, this given piece of DNA, and transcribe it, and we're going to go this direction because it's the top strand, it would look like this. The RNA will be in blue, because RNA is always in blue, GAA. Now, thymine is not found as a ribonucleotide. It is a modification. The base is uracil, and so it's written as a U. And so all you do is take the top strand, turn the T's into U's, and you're good. Now, from an actual practical perspective, T, U, who cares? When you're using a computer to decode a DNA strand, the computer just does it for you. If you were to be all technical about it, yeah, you got to put a U in. I could, and you may find a really, frankly, asshole multiple choice question in which you'll be given a strand of DNA and say the RNA is going to look like what, and then if you write the top strand with T's, oh, you get 2 points off because there is no thymine in RNA. Shame on you. Yeah, whatever. Okay, fine. It's uracil. In terms of extracting the information, T, U, nobody cares. Because mainly, I'm going to have you do a decoding by hand, but I never do that. I just go to the web and use various kinds of DNA manipulation tools, or a program called SnapGene that I've been using a lot, that does all this for you. Yeah, but doing it by hand is actually a useful thing. You've been using these kind of things?
Last semester with genetics.
Or were you at U of I?
No.
Oh.
Here. Genetics
Oh.
with Dr. Smith.
Oh, right. Yeah. Okay. Later in the semester, the biotechnology group at the U of I is going to, I think, do some sort of Zoom, Moodle, something or other. For the past 4 or 5 years, every summer, 2 people have been invited to go to U of I over the summer and work in their genome sequencing facility, primarily working on yeast genes. There's a professor there, Lois, I can't remember her last name. Her daughter played basketball at Millikan, and she was watching a basketball game and happened to be sitting next to Will Coxon, and they started talking about biology stuff, and she goes, "You guys should join our NIH grant." And so they do a ton of DNA sequence analysis. If you know Max Phillips, ask him about it, and he can tell you all about it. All right, so this is what you do when you're planning a molecular biology experiment, is you sit there and stare at DNA sequence and mutter to yourself, and that's how it goes. All right, so now, from this, we can further define these strands. The top strand is referred to as the top strand. Additionally, because this is the equivalent to the RNA that would be produced, it is known as the sense strand. Because when you go to translate it, it will make sense. You actually can get the amino acid sequence from the top strand. Now, the bottom strand is known by a couple of terms. It's called the bottom strand. It's also known as the antisense strand, also known as the template strand. Top strand is also sometimes called the coding strand. I never do that. Anyway, the template strand refers to the fact that when RNA polymerase is actually transcribing this chunk of DNA, the top strand is actually pushed out of the way and told, you go flap off in the breeze, don't bother me. I got to look at these nucleotides. And so RNA polymerase is interacting here, and it is base pairing with the bottom strand to produce the 5' to 3' template. So, in a sense, RNA polymerase is opening up a transcription bubble, making it so it's single-stranded, and getting in, reading the bottom strand, and recreating the top strand. It's just now T is now U. So that is the overall kind of flow that we think about when we're talking about RNA and its relationship to DNA. So these terms, top strand, sense strand, bottom strand, antisense template, that's a real critical thing to always keep in mind. And just to say, check your book, and I posted the figures. I think I actually posted some of the figures from this chapter as well. So we're in chapter 7, and figure 7-6. So just for completeness, top strand, sense strand, also known as the coding strand. Although you never refer to this as the non-coding strand. All right, so that's the organization. This is a really critical point. Now, from here, we can begin thinking about how RNA can be generated using RNA polymerase. And just looking at here, I don't throw nothing out here. All right. So, when we start thinking about the production of RNA, this is stuff that started happening, back in the late 50s, early 60s, and it really took off in the 60s. By the time 1970 rolls around, the genetic code has been figured out. And we'll get into that when we start talking about protein translation. But the mechanisms behind gene expression are starting to become fairly clear. And this is initially done in bacteria. And happily, bacteria, prokaryotes, and eukaryotes share a tremendous amount in common. Now, they do have very specific differences that are like perfect exam questions, but the similarity is just amazing in that the flow of information in living organisms on this here planet pretty much always goes in this standard way. I mean, there's lots of different viral exceptions and stuff, but what this means is you can take a bacterial gene and you can express it in eukaryotes if you monkey around with it, and you can certainly take a eukaryotic gene and express it in bacteria if you monkey around with it, because the genetic code is the same. And that's a very cool thing. And the mechanism is the same. So a lot of the initial work in understanding how this stuff comes about was being carried out in bacteria. So what we're going to do is start talking about the mechanism of transcription. And we do this by going through the prokaryotic system first, and then comparing that to the eukaryotic system. And there's a lot of similarities, but a couple of key differences that we will be getting into. So, in order to make an RNA molecule, we have to have the DNA chromosome, what we refer to in the biz as the DNA template, double-stranded. Then we have to have the enzyme RNA polymerase. Now, in the bacterial system, there is a single RNA polymerase that generates all the different RNA types. And we're going to see that's different in eukaryotes. But just as a little point here, RNA polymerase is making messenger RNA. That gets translated into protein. So not all RNAs are messenger RNAs. We also need to make what is known as ribosomal RNA, and this becomes a major component of the ribosomal RNA. And the ribosome is, I think, considered an organelle. I just call it a ribosome. It is a very big protein RNA particle, a ribonucleoprotein, made up of multiple subunits, lots of RNA in there. The bulk of the RNA in cells is ribosomal RNA. Like something like 90% of the RNA is ribosomal RNA. The other RNA is known as tRNA, transfer RNA, and this is going to be used specifically in protein translation. So, in the bacteria, one RNA polymerase does it all. And in the bacteria, the RNA polymerase is made up of a collection of proteins. It's not a single protein. And it's referred to in the biz as alpha 2 beta prime. This is a shorthand to note that there are two identical alpha subunits and a beta subunit and actually a different protein known as the beta prime subunit. And just as a little note, the RNA vaccine that is being made by Pfizer and Moderna, they're using a RNA polymerase that is isolated from a virus, from a bacteria virus. And the advantage there is bacteria viruses have an RNA polymerase that is a single protein. And so it's a lot easier to manipulate. We use routinely an RNA polymerase known as the T7 RNA polymerase. And you can just buy this, comes in a tube. And it is a single protein, so it's actually very easy to manufacture. And it makes a ton of RNA with a given DNA template. I mean, if you know that you got to make RNA from, to start generating your vaccine, that is an easy step. You take a little bit of DNA template, throw in a bunch of nucleotides and buffer and salts and T7 RNA polymerase, and let it sit at room temperature for a couple of hours, and you just make a ton of RNA. So it's a fairly easy thing to do. You got to purify it and all that stuff, yeah. And then, just find somebody to inject it to. But in terms of its manufacturing, using a viral RNA polymerase is a piece of cake. So these are the different RNAs. Now, just to throw in here, we have a range of different small RNAs, and I'm about to write them up. They're primarily known in eukaryotes as small nuclear RNAs. We'll pick up those in just a moment when we get into the eukaryotes. But for our purposes of bacteria, this gets it done. All right. So this is the RNA polymerase. The next thing we have to look at is how the RNA polymerase knows where a gene is and how it's able to actually transcribe a gene. So if I was to ask you to define a gene, you've all had genetics. You would all be easily able to give me a definition. Honestly, I have to think about it and go, what's a good definition for a gene? It's actually not just falling off a log, obvious. A gene is a portion of DNA that is transcribed. That's my term for it. So, given that, a gene, a discrete region of DNA that is transcribed. And so we next have our next set of diagrams and terms here. When we draw a DNA gene, drawing sort of a cartoon of it, rather than drawing double strands and stuff, this is typically how it's done. It's either done as a line, or sometimes I'll draw it as a box if I'm feeling creative. And we can say, all right, this here's a gene. We can break this down into two parts. We've got what is known as the structural gene, and this is easily defined. This is the transcribed portion. That which is turned into RNA. Now, that is the gene itself that is going to be translated, but there is more to a gene than just the structural region. So a typical way this is diagrammed is with the arrow denoting what direction you're going, and then the number +1. That means that this is the first nucleotide of the transcript. But we've got a upstream and downstream, which are the terms we use now that we have +1, regions. Directly upstream is a region of the gene, DNA sequence, that is known as the promoter. And in more generic terms, we can refer to this as the control region. This is the DNA sequence that is going to actually control the expression of the gene. So in eukaryotes, we spend a lot of time and energy regulating gene expression very carefully. I tend to sort of look down my nose at bacteria, which is, we have a sort of a love-hate relationship. They know that I can always autoclave them, and I know that at any moment they can give me diarrhea. So, we just have an agreement. The promoter region and control region is the portion of the gene that is actually turning things on. Say, in bacteria, I tend to think that they actually have every gene possible slowly trickling out at maybe very low levels. And then when they get ready to really go, they'll start cranking it up. And so bacteria don't have great self-control. They are constantly drooling out things, and you're going, what the hell are you producing that? And it's like, well, it's a very small RNA. RNAs have a really short half-life in bacteria, so you produce a little bit of it, it gets chewed away. And it's only when something cranks out really fast and hard that it starts to work. But the promoter is a critical spot. Now, we can define the promoter in a couple of different ways. A really good definition of the promoter is the spot where RNA polymerase binds. So the promoter is the RNA polymerase binding site. And not surprisingly, there are sequences within the promoter that bind to the RNA polymerase specifically. So the RNA polymerase isn't binding ready to roll everywhere. It is binding at the promoter in a very specific way. Now, that said, in the prokaryotic world, the promoter, or the RNA polymerase, in this form, alpha 2 beta prime, it exhibits what's known as loose, i.e. non-specific binding to DNA. In order to get the RNA polymerase to bind to promoters, we have to add one more chunk, one more protein that is known as the sigma factor. So, we can draw or diagram RNA polymerase in the bacteria in two ways. We can have the alpha 2 beta prime, no sigma factor. This is known as the core enzyme. And then we have alpha 2 beta prime sigma. And this is known as the hollow enzyme. Why the distinction? I will tell you. And that's because without sigma factor, the RNA polymerase doesn't recognize promoter sequences. It just binds sort of everywhere in a very loose way. When you add sigma factor, it now binds specifically to the promoter. So, what we can do is scan through genomes of bacteria and identify a promoter sequence. This becomes a computational thing where we know, okay, here's a gene. This is the first nucleotide of the RNA. We can identify that because you can look at the RNA being produced. And you then look at the sequence just upstream, and we can define what we can think of as sort of the average promoter sequence. And this would be done, sort of statistically, where you develop by looking at a bunch of different DNA sequences, what we refer to as the canonical promoter. That is, it has the consensus sequence. And so you have a bunch of promoters, you look at them, and you determine, all right, what's the consensus? What's the average nucleotide at this spot, And this is how we can determine and generate what we think of as the standard basal promoter, the consensus sequence. And when we do this, it turns out there are two spots in the promoter that are always there, because there's regions of the promoter sequence that are all over the place. It can be a variety of different nucleotides. And it's like this. So we'll put +1 here. So this is the beginning of the structural gene. And there is a very good consensus site 10 nucleotides away at -10. And this is a TA rich region. It's sometimes TAATA or TATA, known as the Tata box. One of the thoughts here is that this is the spot where the RNA polymerase is able to open up the double-stranded DNA. Being AT rich, because AT nucleotides only have 2 hydrogen bonds, GCs have 3, AT rich regions are just thermodynamically easier to open up. That's why we have them there. And then a little bit further upstream, and yes, I'm having to look to make sure that I don't mess it up with the book. This is what I was going to say the -35, but I want to make sure. We have the -35 site is another consensus chunk. And you don't really need to memorize the sequence, but we should know that -10 is Tata, -35 tends to be what I think of as the cat box. It's CAAT. This is the spot where the RNA polymerase is actually making its initial binding. All right, so in the absence of sigma factor, with no sigma factor, the RNA polymerase is exhibiting this loose, non-specific binding. But when sigma factor associates with the RNA polymerase, we now have a very high affinity binding to promoters. So the sigma factor is floating around, and when we have the need for a gene to be expressed, it'll interact with the core and kind of move around, slide around, and bind onto promoters and just start cranking out RNA. So what we find then, when this is studied more carefully, is that holoenzyme, that is with sigma factor, this is necessary for initiation of transcription. This is going to bind the promoter and initiate transcription. Turns out that once you get about 50 nucleotides down the road, you start polymerizing RNA, sigma factor is no longer needed, and it actually isn't even found, and we generate then the core enzyme. The core enzyme is what's carrying out what we know as or call elongation. So, in order to be better, more alliterative, begin transcription, how about we refer to this as transcription initiation. Initiate. So we have transcription initiation requires sigma factor. Transcript elongation doesn't need sigma factor, just the core enzyme. This will play a role here when we get into some gene regulation stuff. All right. That's enough for right now. We'll pick up on Wednesday and begin making an RNA and then compare that to how the eukaryotic system makes RNAs, and we'll see all kinds of cool things. All right. That's all I know. Rock on.
No lab this week?
Nope, we're not going to do lab yet.